Thanks a lot for the introduction, and well, thank you for the opportunity to talk and to interact. Maybe one of the good things, good outcomes of this crisis is that maybe we have more remote interactions of that kind. So today in this presentation, I'm going to discuss how we can make a good use of the data we've been collecting in functional neuroimaging now for decades, using data science and AI techniques. So towards universal representations of cognition. So let me start by caricaturing a little bit the job of cognitive neuroimaging. So let's assume that as a neuroscientist, you have cognitive theories that you want to probe, for instance, on language, how the brain processes language content. And to do so, you will typically select some stimuli that you will present to a participant in an experiment. You will organize the stimuli in an experimental paradigm, and then you take the fresh brain of a 20-year student of the next university, you scan him or her, and you collect the data, you pre-process the data, you analyze them in order to get brain images like these activation images synchronized with the presentation of the stimuli. And then comes the time of statistical analysis, where you try and find statistical relationships between the task performed by the participant, the stimuli that were presented, and brain activity. So I will call encoding the mapping that goes from the task or stimuli that were used in the experimental paradigm to brain activity. So how well these tasks explain brain activity. And decoding will be the converse operation, where you try to characterize the behavior of the subject or some information about the stimulus that was used based on brain activity. Somehow these two things are addressing the same question, but with different statistical models. So I will use both in this presentation. So now to get a bit more background, you all know that many experimental sciences have faced a replication crisis in the recent years. So we have realized that many of our findings are not reliable enough. And especially so in brain imaging, where people have been using small samples to collect data and so obtain relatively weak results from a statistical perspective. And the good thing that people have realized that, so this dates from already 7 years, this paper that I think everybody has read, and people have been changing. They have been improving their imaging practices. They have been increasing the average size of the study. And also they realize that they need to share data because you will never be able to collect enough data to probe all the cognitive theories you would like to probe. You probably need to compare your own data with the data collected by your colleagues. And for this, we need to share data. We need to be able to reuse the data collected by the community. And these things are happening thanks to resources such as NeuroVault, where people can upload the statistical results of their study, or Open Neuro, where people upload typically the raw data. And then comes the next question, which is, okay, now we start to have a pretty rich common resources. So how do we use them? It's actually a bit trickier than we could think, because when you read neuroscience publications and you look at the product of different brain imaging experiments, it's very hard to compare these products because each neuroimaging result corresponds to a way of asking a question, of framing a question about cognition. And each lab has its own culture, its own way of framing questions. which means that it will not be easy to compare, for instance, the results that come from Boston, from Martinos Center, to those that come from Neurospin, because cognitive neuroscientists have different traditions, different ways of probing brain systems. So we need to find a common conceptual framework that makes it possible to leverage the data acquired at Neurospin in Boston and in all other places. And one of the rules here is that you cannot play 20 questions with nature and win, which means that somehow we need to simplify the questions, because each study typically addresses a very specific question, and it is hard to generalize these findings. So somehow we need to simplify the questions to find a common framework in which the questions are slightly less specific, but can be looked at with different data sets. So I'm giving references because these two papers have discussed this question quite in depth. So what I'm going to do now is to present you two ways to frame questions and to analyze large data corpora. One will be what I would call joint analysis, where you analyze jointly many different data sets, and you will see that large studies inform the analysis of small studies and improve the accuracy. It's a kind of transfer learning if you frame it in terms of machine learning. And so essentially the mechanism will be to leverage joint representations across studies. And the second way will be to to identify semantic commonalities across studies and use them explicitly. I will call that some mega-analyses. And the difficulty here will be to define a common vocabulary across studies. So let me first explain what I mean with a joint analysis framework. So we have started by leveraging the data that are available in open data resources such as OpenNeuro. So if you look at OpenNeuro, there are several tens, I think several hundreds even of data sets. What we have done was to analyze some of them with the collaboration of the OpenNeuro team. And actually there are some resting state data, in particular the very rich data of the HCP dataset, and also some data related to cognitive protocols. I listed different studies. In our case, we have used data from about 40 studies. So it's 40,000 task-fMRI contrast maps that have been used. And what we do with all these data is to throw them into a big, say, a big neural network that will be used to pre-process them. But this neural network actually has a very simple structure. The first layer of the neural network is just a dimension reduction, which is learned on the resting state data only. So just using the HCP resting state data, which are so huge, we can learn simply a decomposition of the brain into territories. In that case, 500, but we could do even a bit more. So in today's experiment, I'm talking about this 500 regions decomposition. And then we use that to essentially simplify all the other images. So we can represent each of these images by the values that they have in these 500 regions. So instead of a big image, you just have a 500 dimensional vector. It's much simpler. So actually, you can have a reference here about an atlas that has been published recently using this approach, and we show that this representation is very powerful for many brain imaging tasks. But then comes the supervised part. You've got these 500 dimensional vectors, and from that, you would like to be able to classify, given a brain image, classify it according to the functional contrast that it represents. So here I'm talking about fMRI contrast, of course. And the idea, for instance, is to be able to say whether an image represents, for instance, a zero back face memory task, working memory task, or two back place a memory task. For instance, if I take this example of from the HCP dataset, but this for all possible contrast of all studies. And the structure of the network is to further reduce the representation from 500 to 128 using these bottleneck layers, layer in the middle, and then finally have classification heads that classify within each study the contrast to the category it belongs to. The idea is that these two layers are trained jointly. So the bottleneck layer, the middle layer, is actually informed by all the classification tasks. It's a supervised model. So to put it slightly differently, you start with your images with 200,000 voxels. You reduce them to the loadings on a 512 resting state network decomposition. Then you finance this decomposition by creating 128, say, task-optimized networks. And from that, you can try and predict the category of the stimulus or some information of the stimulus that was presented. First things to do then is to look at the accuracy you obtain by doing that. So the prediction accuracy, the accuracy of the task can be computed for each of the data sets. So you've got the, I think, 35 data sets that have been presented. And you see with the black bar, the accuracy that was reached by this classifier, which is compared with the accuracy you would reach with a standard classifier in gray, a classifier that would only know that particular study. So that would only take the data from that study and classify, try and classify the images. And what you can see is that overall the black bar is longer than the gray one. So we have a gain in accuracy, actually quite impressive gain in almost all studies. The average gain is about 6%, which is non-negligible. And of course, we have a loss in a few studies, but in 80% of the cases, we have a gain in accuracy. If we look a bit more at the study, so we can compare the accuracy gain on the y-axis here to the baseline accuracy that we had before doing this, this joint analysis. And we see there is not much influence. Of course, when you're already very accurate without the joint modeling, you don't gain much. You gain more if the accuracy was already better than chance, but like about 70, 80%. Maybe much more interestingly, you can see on the right that the gain in accuracy decreases with the number of trained subjects in the study. So the smaller the study, the more you gain by learning a joint representation from this study together with the other. So there is some transfer from the large studies to the small studies. And finally, the third set of bars here shows that using the resting state representation or dimension reduction only does not buy you any gain. Essentially, it's the same level of accuracy as classifying from the initial images. Where you really gain is with the supervised representation that tries to classify all the studies jointly. This is where you gain. A further word on the transfer between studies. You can take some of the larger data sets and see what happens if you redo the experiment, but by decreasing the sample size. It's like a learning curve in machine learning. And you see that, of course, when you decrease the sample size for this particular study, accuracy decreases, but the multi-study decoder in a range still has a better accuracy. And actually the gap between the multi-study decoder and the standard decoding increases when the sample size is smaller. So, which means that there is even more transfer. So it's very logical if you think in terms of Bayesian, for instance, Bayesian priors. The less data you have, the more you need to rely on the prior. And so the more you will gain by relying on the good priors. So this is exactly what these curves illustrate. One feature of this neural network is that it has no non-linearity. It's still a completely linear model from the beginning to the end. So the classification rule that you use in each study is a linear classifier. And so as a linear classifier, it is pretty easy to interpret the discriminative patterns. And actually, you can look at the intermediate representation that was used, the 128-dimensional representation, and extract dimensions from it together with the tasks that are well discriminated by this representation. And so you have, for here we have illustrated, I think, 12 such components, together with a word cloud that represents the tasks that were well discriminated by this component. So now we can really relate some topographies together with the cognitive conditions that they can discriminate. So it's a kind of functional atlasing, if you like. Of course, we can uncover some usual suspects, and maybe sometimes some slightly more sophisticated ones, like calculation, for instance, is interesting, or the one on false belief story, or these experiments on risk taking, for instance, in the frontal lobe. So altogether, so this makes an atlas that we are now sharing on the CogSpaces site. And so this is currently submitted work otherwise, but you can essentially access all the resources already freely. I think I can stop here. If there are some questions on this first part, I'm happy to answer now.
It seems like there are no questions. So
okay.
If anybody has any questions.
I hope I was clear enough. Don't hesitate anyway to ask. Anyway, I'm going to go on. The next topics are quite related. So far, what we have shown is that we can find or we can design a framework in which we jointly analyze many different data sets. And this is good because these data sets can share common representations, and this is beneficial to the accuracy of your models. Well, but so far the relationship between the different data sets was implicit. There was no, at the end, each data from each data set were classified independently. Here now we try to really explicitly model the commonalities, the semantic commonalities between data sets to improve cognitive mapping. So once again, it's just a different representations of the same ideas as previously. So you have brain maps with labels. Now we are typically considering situations where you would analyze, for instance, the NeuroVolt data. I don't know if you're used to looking at NeuroVolt, but it contains now about 100,000 fMRI activation maps together with some tags and tags or labels that characterize
Bertrand?
Yep.
Yeah, can I interrupt you? There is a question. So I'm gonna read it. The interpretability of the linear network is helpful. Could you please say a bit more about what might be the trade-off relative to a network that includes nonlinearity?
So I think, I'm just trying to remember, I think we have tried quite hard to increase prediction accuracy with nonlinearities, and we completely failed. I think that the reason deeply is that the signal-to-noise ratio of the classification problem is pretty low, and capturing the effects with the linear models is still powerful enough to do the best that you can achieve. So at the moment, having a linear model is a sensible choice in terms of prediction accuracy. And it's a huge benefit in terms of interpretation because we know that interpreting the weights of a nonlinear classifier is very tricky. There are lots of ways to get it wrong. So it's fortunate, I would say, that for the moment and for the amount of data that we have, it's still a good solution to rely on linear classifiers. Okay, we can maybe discuss that further. So let me get back to my NeuroVolt now, maybe. Okay, so you've got the labels and the images. The encoding analysis tries to find a mapping from labels to images or tags to images. And the converse analysis decoding tries to infer the tags that might be associated with an image. The difficulty here is that it's not a multi-class problem with a mutually exclusive class, as we do traditionally in supervised learning. It's a multi-label problem. And an image can have different labels. They are not mutually exclusive. So there's a combinatorial set of possibilities to label an image. So we rely really here on NeuroVolt. When we did this experiment, we could, there were about 70,000 images that were available in there. And there were among them 48,000 that we could use for the decoding. So that would survive some basic quality checking. We take only human images and that have reasonable values that we can at least figure out, etc., etc. And so this gave us 48K fMRI maps. So this is for the imaging part. But you will see that increasingly we will have to think of how do we annotate these images, and for this we need a knowledge base. So for this we are relying on the Cognitive Atlas proposed by Paul Drac Lab in Stanford, that essentially contains concepts and tasks. Concepts are cognitive concepts that describe brain states or kinds of brain activity. You could, for instance, think that working memory is such a concept. And you have tasks, which are some classes of paradigm used to probe the concept. For instance, a two-back task would be a way to probe working memory. So two-back is a kind of task. Okay. So if you do the analysis in the encoding direction, you take a certain concept, a certain cognitive concept, and you try to see what is the brain correlates of this concept. So to do that, you take your collection of images, X, you create a design matrix that represents the occurrence of the concepts for each of the images in NeuroVolt. So the number of rows will be the number of images that you have in NeuroVolt, and the number of columns will be the concepts. And you try to with for instance, a linear model, you try to capture the terms effect per voxel. The problem with that is that so we did that a few years ago now with a relatively smaller data set, and we had this problem that some of the terms will be highly correlated or anti-correlated. For instance, visual and auditory will be very anti-correlated because you typically use one modality or the other, but not both. Or like count and digits will be very highly correlated, which means that it will be hard to disentangle the effect of counting versus the effect of digits, and so on. So of course it works somehow. You still have some issues like auditory gives you auditory regions, but also some visual regions in negative due to the anti-correlation in the design. Count is okay with lots of visual activity, but why not? Button press is funny because of course you obtain the motor cortex, but the left motor cortex, which means that implicitly in most experiments with button presses, you use the right hand, or at least right-handed subjects. So you've got this lateral bias in our data. But there are some examples which are less nice. For instance, abstract patterns and shapes will provide almost the same map. So the encoding analysis is not very powerful at disentangling the nuances between related concepts. So to solve this problem, we need to formulate the question the other way around. Can we decode an image? So the name of the game is that you give me a brain activation image and I try to guess what the task was, what the person was doing. So at least I try to guess some labels that could be associated with this image. So it's a multi-label classification problem. So we have done that. You can see the related publications here. The first attempt was on 20 categories that were hierarchically organized. So 20 categories is very low to summarize all what we know on cognition, but I told you from the beginning that we would have to simplify our questions. But we could show that with our decoder in blue, we could obtain a relatively high accuracy, slightly better than different alternatives. And for instance, better than what, for instance, using simply neuroscent would obtain. That was great. And even better, at that time, we were also using linear classifiers. And with linear classifiers, you can look at the support of the prediction. And so for the 20 categories, there were like 9 visual categories, some motor categories, some auditory and language categories, and finally, saccades and mental arithmetics. So for all those, we could obtain some relatively relevant brain territories. Of course, we all know these mappings. I'm not saying that finding these regions is novel. The great difference is that you know that these regions are involved in this task because you've read this from the literature. No, I show it from you, and it's coming from the data generated by this community. So this is grounded in actual data. But we want to go further. Now we want to extend the number of concepts. We want to go essentially into the wild with a much larger set of concepts. So to do so, we have redone this with the whole NeuroVault collection, which I have described previously. And the first thing to do, so from NeuroVault, we extract the brain images. We select the good maps. We pre-process, for instance, by standardizing them. And then we compress them by taking the loadings of the images on a certain dictionary of regions, as I did previously for the joint decoding analysis. And with this, you can essentially simplify an image to just a vector of 500 or 1,000 values that summarize this image. This is the easy part. The difficult part is with the concept representation. So people provide some tags on NeuroVault when they upload images, but these tags are very noisy because they represent the intent of the person who generated the data. They may not be consistent across groups. So we first try to match these annotations to some concepts that are found in Cognitive Atlas. And then we found that we would have to enrich this to obtain a set of labels that we would try to infer from the multi-label decoding. Doing this by just using the concepts that we could identify from the annotations, we could do a simple validation on the data. So using about 10,000 images for validation and training on the others. And with that, we were able to test 37 concepts, which are listed here. And you get on the right the accuracy that we reached for these different concepts. And you can see that for many of them, we do much better than chance. So actually, what is plotted here is the AUC. So chance level is 0.5. And you see that in 80% of the cases, we do clearly better than chance, which is great. So we can decode many of these concepts. Actually, we do better, for instance, than the GCLDA, green, a model that was proposed by Yarkoni and colleagues a few years ago to do the same kind of tasks. And that was trained on the Neurospin database. So we could use this model. And we see that by leveraging the images, we obtain higher accuracy than by using the GCLDA model trained on the Neuroscent database. On the left side, what you see is the frequency of the terms across images. And the surprising thing is that the most frequent terms are the bottom. These are the terms that are harder to decode. So, which is quite counterintuitive. I would have thought the converse. But it seems that some of these terms are used quite often to describe cognitive conditions, but maybe in a slightly inconsistent ways across experiments, which means that these labels are very noisy. So essentially, they become hard to identify. For instance, working memory, surprisingly, is hard to obtain, or response execution, which is pretty vague, of course, is hard to identify. Okay, so this was 37 concepts is good, but it's not good enough. And what we observed is that actually the main issues was in the labeling. And of course, we need to do some curation on the labels. And especially a very simple curation is just to take into account the logical structure between labels, like the hierarchical structure. For instance, if you've got a speech perception label for one image, it probably involves voice perception, which then probably involves auditory perception, or also known as audition or listening. I think these things cannot really be distinguished in our case. But certainly when we have speech perception, we could also use these other labels. And so doing this, we could just simply enrich the set of labels by using these inclusion rules. And when we do that, we can see that now we can probe many more labels, a bit more than 50, which is more than previously. And actually the accuracy increases, which means that we can decode much better these labels. Which is great. So essentially, we have removed some false negatives from our training set, and we have now a more powerful classifier. And at least for all these terms, we can say with confidence that we can identify them from the NeuroVault database, which is a great progress. We can still actually, that time, for the first time, we could use a nonlinear classifier, and that obtains slightly higher accuracy than the linear classifier. Difference is not very large, but it's there. But still, we can, for instance, take the best fitting linear classifier and look at the map, or look at the sensitivity map for a nonlinear classifier and interpret them. It's always useful to open the box. And of course, some of the discriminating patterns are completely uncontroversial. They're almost trivial, okay? For instance, left toe decoding is trivial. More interesting, maybe, if you compare the encoding and decoding maps for a given term, like syntax, is a pretty abstract term. You see that just doing the encoding, so looking at the brain correlates of encoding, you will see pretty wide regions along the STS. But if you try to decode it, then you have a different patterns with, it's more the anterior STS, which will be useful, at least that portion of the STS, and the inferior frontal gyrus on the left hemisphere. So you see that decoding still provides probably more specific information than standard encoding. And there are some cases which are more worrying. For instance, when you look at face perception, it's great. You see the FFA that comes, and so face perception is decoded by FFA activity. Good. Now, if you do face recognition, the patterns is similar, but with the top of it a small spot in the motor cortex. It's like if face recognition was just doing face perception and pressing a button. Less interesting. And even worse, if you look at emotion perception, you see that you've done the same pattern, which is just an artifact from the fact that people study emotions by showing fearful faces. And actually, there's this shortcut that much of the emotional images are just face images in our dataset, on Neurovault at least. So this just represents the biases of our community. Of course, that can be solved by adding more data sets that have different ways of testing, of probing this emotion perception concept. Okay, I can proceed with image annotations unless there is a question. Maybe I can go on, and anyway, we can have the discussion later. So, of course, so here I'm really presenting some image-based kind of decoding or meta-analysis, if you like, but we need to compare it to coordinate-based meta-analysis that has been used successfully by the community for decades now. So I will present some new attempts also to mine the scientific literature. And the motivation here comes from our previous experiment. You see that most of the noise in this analysis is not in the data, in the fMRI images, it's in the way we label them. So we need to learn to label data. And so for this, we need to have some simple yet consistent vocabulary or ontology to describe cognitive processes. And of course, we can rely on knowledge bases, but we need to find a way, a balance between what knowledge bases tell us and what data tell us. And so our motivation here was to mine the neuroimaging literature because many observations that exist currently are just stored in text. Like a sentence like that one, for instance, can contain a lot of information on brain activity. And we should be able to leverage this to decode this term. Maybe just when you have such a sentence, we should be able to draw a brain image that represents it because it actually corresponds to brain structure. So our objectives was to transform neuroimaging publications into brain maps somehow to make it simple. So we have done a meta-analysis of text-only corpora. The idea is to have automated meta-analyses such as those performed in Neurosynth. So what we have done was to text the full text of all the neuroimaging corpus that is accessible in PubMed. And so we extract two things from these publications, the text, so all the set of words, and the coordinate tables that contains brain positions. From the text, we can just do some very classical NLP manipulations to obtain a vectorized representations that summarizes the concepts present in a given text. And this will provide us an occurrence matrix, X, that represents in each publication the concepts that are occurring. And on the other hand, you can take the brain coordinates that have been reported in MLI space, and then maybe convolving them with a Gaussian kernel, you can obtain a brain map corresponding to that publication. And the name of the game is to find the beta coefficients that relate the occurrence of the terms with the topographies that you have obtained. It's similar to what Neurosynth does. The big difference is that Neurosynth only uses the abstracts of the text. Here we use the full text, which is much, much richer in terms of semantics. So we've done some experiments and we have shown that using the coordinate information is indeed the best representation. It will give you the best predictions on left out articles, the way to represent them. It's actually better than using, for instance, brain atlases, which are essentially too coarse and not consistent enough across the literature to provide reliable mappings. But more important, and this is maybe the main difference with Neurosynth is that we have started to rely on semantic relatedness. For instance, if you take the term melody, you won't find in the neuroimaging publications enough articles that use melody significantly together with brain coordinates. So if you use melody only, you will do a very poor job at mapping this term because you will have too few publications to map that term. So what you should rather do is to observe that melody is related to music, musical, auditory, and pitch, which are terms that are well mapped across the literature, and essentially use the maps related to these terms. That's what we do. We propose a term, a map for melody, which is a combination of all these terms. And this works much better than using, for instance, simply the mapping from melody to coordinates. It's a kind of bias variance problem. You will accept some bias. You bias melody toward other semantic related terms to decrease the variance and obtain a better predictor. So by doing cross-validation, we show that this works much better. And same thing for meaning, for instance. Meaning is not, it's a very complex term, very polysemic term actually, but you can relate it to other terms that are found more frequently and obtain a map which is reasonable for it. And same thing for agnosia, for instance. So we have run a lot of validation experiments, which I'm not discussing now. You can find all these details in this eLife paper by Jérôme Deques. But more importantly, we provide now a web interface, Neuroquery, so you can find it. And please play with it. So you can type any text. It's not, you don't have a restricted vocabulary. You can just type a paragraph or even a long text, and it will try, it will identify the terms in the text that are mapped with the current meta-analysis, and will provide you a candidate map to map your paragraph, the text that you have provided, which is another difference with Neurosynth that only maps single terms. So this is really a complement to what Neurosynth is providing. In some few queries, Neurosynth will do better than Neuroquery, but we find that for complex queries, at least Neuroquery is more powerful. For instance, another example that I give here is that we have taken several terms related to mental arithmetics, and with Neuroquery, we would map them more or less similarly, relying on the semantic relatedness between these terms. And this is certainly better than just using the occurrences of these terms across the literature, which provides very noisy maps or empty maps in some cases. Okay, so this is to improve the way we describe brain images. And I'm a machine learning person, and you might say, okay, it's easy for you to say to the community, oh, please provide good images and good annotations and just play with this data. So I'm not only analyzing data, I'm also providing some. This is an effort that I would like to share with you to finish this presentation. So we have launched a project, an individual brain charting project funded by the Human Brain Project, which consists in selecting 12 participants that we have been scanning for 5 years now, and we're going to scan them again for the coming 3 years. And we have now they have run 35 different acquisitions. And so we obtain high resolution images. It's 1.5 millimeter resolution, which are pretty high quality. You can see that we are here replicating, for instance, the HCP protocols, and we obtain relatively clean maps on these subjects, together with annotations that are terms from Cognitive Atlas that we use to describe these images to make them useful for the community. So it's a project where we have only few participants, but the idea is to do some very deep phenotyping of these participants. And of course, it's very interesting to look within participants at the consistency of activations across a lot of batteries, a lot of tasks. And so we are analyzing this data, but we're also making them publicly available. But since this experiment is still ongoing, we need good protocols. So if some of you have good protocols that they know are robust and you can rely on them, please share them. So we can talk about that. Please share them so that we can also reuse them, because we would like to do this very comprehensive scanning to help disambiguate the way different fMRI protocols probe the same cognitive functions. So just to show you some very initial analysis of this data, we have tried to create some networks that represent some activities, summarizing here the first 60 contrasts that we could probe. And you can see both maybe the consistency of the patterns across subjects, but also some variability, some of which is noise, some of which may be more interesting. And the idea is to cross-validate again and again these mappings with new protocols that can be used so that we can use. These data are publicly available in NeuroVault and OpenNeuro. Of course, since the dataset is pretty small, we have made sure that we were reproducing the contrast of some other large-scale experiments. For instance, we have reproduced the contrast of the Human Connectome Project study, and we show that we obtain a good similarity, with the exception of the reward task of the HTTP, but which is not to work very well, or something for other batteries. And for instance, we can start to show that we can play the game where we try to predict the contrast from a new experiment given the contrast from a previous task. And we do this much better than chance overall. This is an illustration. We just published a paper in Human Brain Mapping to show these effects. So I think it's time to conclude now, to have time for discussion. So I've tried to show you that we could use both decoding and encoding to obtain a better specificity in brain mapping, which is a global goal of this work, the global direction of this goal. Maybe the main roadblock here is that finding commonalities across cognitive studies is hard. And we are working on solving that. So what I presented today was big data approach, which is not, I know that cognitive neuroscientists don't always like it, but I think it's at least a way to go to better leverage the data that we have already generated collectively. And the general philosophy of this approach is to extract weak signals from a huge amount of data and to find common representations across data sets, which I also call the bottleneck in the neural network model. Of course, there are lots of ongoing work, especially on identifying the structure that underlies the cognitive concepts to be able to better label images. Another point which is very important to me is that between good ideas and useful work, there is software, and you know that perfectly well. So my team has been contributing many Python software. So first scikit-learn for generic machine learning tools, but Nylearn for fMRI data analysis, and MNE, which is actually a common baby between Neurospin and Boston. And of course, these software are open, and please do not hesitate to interact with us on the improvement of these tools. Finally, I would like to acknowledge the contribution of my team and of many collaborators on this project, especially those who have contributed these huge data repositories, which are so useful now. And thank you for your attention.